Hello everyone, this is Ola. Welcome to the second video of the muscle physiology. Here we will discuss the mechanics of skeletal muscle contraction. So when the action potential goes along the skeletal muscle cell membrane, which is the sarcolemma, this action potential triggers calcium release from the storage place, which is the sarcoplasmic reticulum. Now, calcium here attached to the troponin, and this leads to movement of tropomyosin away from the myosin binding sites on actin. Now, actin is not covered, and the binding sites on actin are exposed. Myosin loves actin, and here's a chance. Here it is not covered with tropomyosin. Then here the myosin head attracts the actin toward the center of the sarcomer. And here ATP must be utilized as the source of energy. What happens here to the sarcomer? So here is a relaxed sarcomere, and the A band is the whole length of myosin. Here is the H zone, which is the part in the center of the sarcomer that has only myosin with no overlapping actin. And here at the sides, we have the I bands, and they have only actin. So when the sarcomere shortens during the skeletal muscle contraction, what will happen here? So you can see that the A band stays the same. It does not shorten. So A band does not shorten. How about the H zone? The H zone is very narrow. How about the I band? Almost disappear. So during skeletal muscle contraction, the A band stays the same, H zone becomes narrow, and I band becomes narrow. It almost disappears. Let's go here step by step with the sliding filament theory uh, that leads to skeletal muscle contraction. So here is a resting skeletal muscle. You can see that the actin is covered with the tropomyosin. Troponin is fixing tropomyosin in place. And here is the thick filament myosin. This is the myosin head. It has two sides. One side to attract actin, the other side is for ATP. This part between the head and the thick filament is the cross bridge. Now, during contraction, the first thing that happens is that calcium unlocks troponin. Tropomyosin moves away, and now the myosin binding sites on actin are exposed. So what will happen here is that the myosin goes to attach to the actin, the myosin binding sites on actin. So here, when the actin binding sites for myosin are exposed, are not covered, then the myosin head attach there. And you can see here that a phosphate is dislodged here from the myosin head. So here, during relaxation, you have ADP and phosphate. Both are linked with the myosin head, but as the myosin head goes to attach to actin, the phosphate goes away. Then what happens when the ADP also goes away? So here the myosin head doesn't have doesn't have the phosphate nor the ADP. Then in this case, the cross bridge happens and actin is attracted by the myosin head to the center of the sarcomer, which is the contraction. Here the sarcomer shortens. And during this time, another ATP, this one, is trying to go again to attach to the myosin head. We call this power stroke. So during the power stroke, actin gets pulled to the center of the sarcomer, leading to muscle contraction. Now, when ATP goes to attach again to the myosin head, at this point of time, the myosin head leaves actin. Now, ATP is hydrolyzed again to phosphate and ADP, and the cycle repeats. Then, when the phosphate goes away, leaving only ADP, now the myosin head is attached to actin, and again the cycle repeats. We call that the cross bridge formation or the sliding filament theory. So during skeletal muscle cell contraction, what happens in a summary? First of all, we have acetylcholine is released at the neuromuscular junction, leading to opening of the ligand-gated sodium channels. Action potential then happens. The action potential triggers the release of calcium from the sarcoplasmic reticulum. Calcium now unlocks troponin and tropomyosin moves away. So now actin is not covered. Then linkage happens between actin and myosin, and then myosin attracts actin to the center of the sarcomer, leading to shortening of the sarcomere. And this is muscle cell contraction. Then what happens during relaxation? First of all, acetylcholine in the synapse is broken down by the acetylcholine esterase enzyme. No neurotransmitter is now in the synapse. Then no action potential goes along the sarcolemma. Now, calcium ions will go to be stored in the storage place sarcoplasmic reticulum. And then the linkage between actin and myosin break, and actin and myosin filaments slide apart. So now we have the relaxed sarcomere. And then this is relaxation. So you'll see that the I bands become longer and the H zone expands during relaxation. Okay, now with some review questions. Let's place the events involved in muscle contraction in order. What happens first? What happens first is that acetylcholine is released into the synapse and it stimulates the nicotinic receptors. So here, a nerve impulse causes acetylcholine to be released at the neuromuscular junction. Then acetylcholine binds to its nicotinic receptors in the sarcolemma, and this triggers calcium release from the sarcoplasmic reticulum. Calcium unlocks troponin, tropomyosin moves away, and now actin is exposed. Then the myosin attracts actin to the center of the sarcomere and utilize ATP for that, which is the source of energy. This is skeletal muscle cell contraction steps. The area of the muscle fiber where actin and myosin myofilaments overlap is, here we call that the A band. What is the region of the sarcomere that contains only thick filaments, myosin? It is H zone. During muscle contraction, the actin myofilaments slide toward the center of the sarcomere, so they slide toward the H zone. That's why the H zone becomes narrow, because the H zone has only myosin. When actin goes to the center, the area that has only myosin becomes very narrow. Do thick and thin myofilaments themselves shorten upon muscle contraction? They do not. What happens is that only actin moves to the center, but it does not shorten itself. What do we call the distance from a Z disc to another Z disc? This is the sarcomere found in the myofibrils. During contraction of a muscle, calcium ions bind to what protein? They bind to troponin. So tropomyosin moves away, and now actin is exposed. What happens during the power stroke? Here, ADP is released. So now the myosin head does not have phosphate nor ADP attached. Then it goes and then attracts actin to the center of the sarcomere. The bond between the actin and myosin head is broken when ATP binds to myosin head. Now the myosin is released from actin. The sequence of cross-bridge formation and myofilament movement will be repeated as long as we have calcium. As long as we have calcium and ATP, this can be repeated. Once contraction stops, what happens to unused calcium? It has to be stored back into the sarcoplasmic reticulum. What is the source of energy of bonding myosin and actin? Myosin needs to utilize ATP as the source of energy.